ट्रांसलेशन की एक नई क्विज में आप सबका स्वागत है. The typical rate of translation in bacteria is about 20 amino acids per seconds. So option C is correct. Here the translation speed is actually being concerned. So the typical rate of translation in bacteria is about 20 amino acids per second. Option C is correct. 20. So it is actually 20 amino acids per seconds. Option C. Amino acid selenocysteine resembles cysteine and encoded by UAG, which is one of the stop codons. Here you can see the UAG, which is actually the stop codon in universal genetic code. It codes for 21st amino acid selenocysteine, which resembles cysteine and encoded by a stop codon of universal genetic code, that is UAG. So the correct answer is option A, UAG, which codes for selenocysteine. Oh, I am really sorry. It is actually UGA. It is UGA. The answer of second question is UGA. Streptomycin, a basic trisaccharide, binds with the 30th subunit of the bacterial ribosome and inhibit protein synthesis. That is, it is the option A, 30S subunit, which inhibit protein synthesis. Chloramphenicol binds with 50th subunit of the bacterial ribosome. That is, the option B is correct. So the correct answer is option B, 50s subunit of the bacterial ribosome. So the correct answer, you can see that the streptomycin binds with the 30s subunit of the bacterial ribosome and inhibit protein synthesis, but chloramphenicol binds with 50s subunit of the bacterial ribosome and inhibit protein synthesis. So the correct answer is option B. Translation involves all the three processes: initiation, elongation, and termination. These were all involves in translation. Actually, every process has these three steps: the elongation, the initiation, elongation, termination. So the correct answer is option D, all. All is the correct answer. Translation involves initiation, elongation, termination, all. Correct answer is option D, all. Activation of amino acids require enzyme amino acyl transfer RNA synthetase and then amino acyl transfer RNA ligase. So correct answer is option C, both. It involves both the enzyme for attaching the amino acids to transfer RNA, and the transfer RNA activation involves the ATP. So activation actually involves both the processes: amino acyl transfer RNA synthetase and amino acyl transfer RNA ligase. So the correct answer is option C, both. Next question is, with a few exceptions, organisms have how many amino acyl transfer RNA synthetase? One for each amino acid. So it has 25 amino acyl transfer RNA synthetase, if I am correct. The correct answer is option C, 25 amino acyl transfer RNA synthetase enzyme, one for each amino acid. So the correct answer is option C, 25, if I am not mistaken. Then the correct answer is option C, 25. It is option B actually. That is why I am saying that I am not mistaken. It is 20, one for each amino acid. I am sorry. The correct answer is 20. Initiation of translation involves binding of 30S, then 50S ribosomal subunit to mRNA, and then is joined by the 50S. The first unit which binds is 30S, and then the unit which binds is 50S. So the correct answer is option A, 30S and 50S. This is eighth question. Option A is correct. Ninth question, the Shine-Dalgarno sequence is complementary to a region at the 3' end of the 16S rRNA. The Shine-Dalgarno sequence is complementary to a region at the 3' end of the 16S rRNA. It is Shine-Dalgarno site or ribosome binding site is actually complementary to a region at the 3' end of the 16S rRNA. So in my views, it is option A. The last question is the first amino acid in any newly synthesized polypeptide is N-formyl methionine in E. coli and other true bacteria or eubacteria. So the correct answer in my views is the option A, but it might be all because the methionine is formed in, but in my views it is actually E. coli and other eubacteria. So option A is correct in my views. This is all about a quiz on translation, which is, thank you.